Assalamualaikum and greeting to everyone, to my SB Dr. Nur Akila Ibrahim and to fellow evaluator. My name is Muhammad Anas bin Ibrahim, meeting number S56501, and today I will be talking about the effect of storage condition on growth of microbiota in commercial fish feed. Okay, the background of study. So fish feed is very essential part in the aquaculture industry. So any prolonged storage, high temperature or humidity condition may facilitate the fungal development and may produce mycotoxin, which will compromising the fish feed quality. The background of study, the humidity and temperature level during storage of fish feed may influence the formation of mycotoxin. Problem statement. So fungal infection on fish feed may reduce the palatability, which resulting in a huge effect of the fish dietary and growth. Significance of study, the storage condition can determine the growth of microbiota. So from this study, we can improve the storage quality of the fish feed. The objective of this study is to evaluate the effect of storage condition on fungal growth, to isolate and identify microbiota of fish feed from different storage conditions, and to screen the mycotoxin from the isolated fungi. Okay, next is the material and method. So this is the overview from my final year project, which is source of feed, moisture content, storage condition, isolation media, isolation of fungi, phenotype identification, and mycotoxin analysis. Okay, we go to the experimental design. So in my project, so I've been using 6 different type of storage, which is dry pellet 1 is a dry pellet, has been stored in a closed cover container. DP2 is a dry pellet, has been stored in the slightly open cover container, and DP3 is a dry pellet without any cover container at all. So for the WP1 is a wet pellet, has been add additional water, has been stored in a closed cover container. WP2 is a wet pellet, has been stored in a slightly open container, and for the WP3 is a wet pellet, has been stored with no cover at all. So first one is the source of feed. So I have obtained the fish feed from the local pet store, which is a Gong Badak pet shop and aquarium, which located nearby UMT. So the fish feed that I've been using is a Star Feed brand, which is a common in Malaysia. Okay, next is the moisture content. The moisture content for this fish feed is 12% moisture. So next is the storage condition. I store the fish feed inside of a sterile styrofoam box that has been stored in a lab postgrad nearby Enos. So for the isolation media, I've been using potato dextrose agar, and the isolation of fungi is in order to isolate the fungi, I've been making a serial dilution before I swap onto the PDA. So after 24 hours, I observe and the fungi form has been inoculated inside of a new PDA, so I can make a series of pure culture. For the final phenotypic identification, there are 2 types of identification, which is macromorphology analysis and micromorphology analysis. For the macromorphology analysis, I've been observed using naked eye. As for the micromorphology analysis, I take the fungi sample and make a staining process so that it can be observed under the microscope. And finally, for the mycotoxin analysis, I prepare a coconut cream agar CCA agar onto the petri dish so that I can incubate the fungi on the CCA so that it can be observed under UV light. Okay, as for the result and discussion, so the first thing is evaluation of fungi growth during storage period. So throughout this project, I've been observed the fish feed sample after 5 days. I have noticed there are no fungus formation on the dry pellet 1 and dry pellet 2, but on dry pellet 3, wet pellet 1 and wet pellet 2, there are slightly fungus growth. As for the wet pellet 3, there are moderate fungus growth. Okay, next is a macromorphology analysis. As you can see on the slide, this is a pure culture sample from my project. So as you can see, this is from dry pellet 3. Okay, the texture of this fungus is a white film coat. So the opacity is opaque, the color white, the form is irregular circle, the diameter is 2.5 cm, elevation is flat, and the margin is filiform. For the micromorphological analysis, this is the fungus sample from the sample wet pellet file. So this fungi has been observed under 1000 magnification. So as you can see from the slide, the fungi is septate hyphae. So you can see the spore is a egg shaped spore. So we can see the microconidia on stalk, and we can see the rhizode. So for the mycotoxin analysis, we can see the fungi growth has been observed under UV light. So on the further left, this is a fungi growth on the CCA agar. So it's shown that it glow orange color when exposed under UV light. Okay, as for the conclusion, storing fish feed in wet condition with no cover will get a moderate fungal contamination. This can be seen within the 4 or 5 days, which resulting in fungal growth, color changing and odor, compared to others with mildly infestation and no fungal growth for dry closed container. Therefore, we can conclude that wet surrounding and open container may heavily affect the growth of fungi. Okay, here's the Gantt chart and here's my reference. Thank you.